We're going to set up a colony PCR. This PCR is going to test for the presence or absence of a transposon upstream from the gene FLHD, which is involved in motility of the bacterium Escherichia coli. Here's a few things. We have some nuclease-free water. We have a PCR master mix, which has most of the components. This is a 2X master mix. And we have our two primers that we're going to use. We have our PCR tubes, and we're going to set up 3 reactions. We'll label our tubes M for modal, NM for non-modal, and then just a dash for our negative control. And so we're going to add our components. I have my protocol sitting right here. And so add our components. We're going to add the water to each of them. So I'm not making a premix. I'm adding individually because there's so few reactions. Okay. And next, I will add the primers. You may have noticed that I use the same tip for pipetting the water. This does not have to be sterile. And so when I'm just pipetting the first component, I use the same tip. But now I'll change tips as I add each component individually. Okay, so I add the PCR master mix last because that has the largest volume and makes it easier to mix everything. You know, mix all my components. I just pipette up and down gently. See that? Pipetting up and down. And then when I'm done adding that one, I close it. So I don't add anything else to that tube. So now we've put all our components, our primers, our master mix, which has the polymerase, magnesium, the DNTPs, and water. What do we do next? So we need a template. And our template, this is a colony PCR, we're going to use colony on a plate. So we have E. coli, this is MG1655, and this is our non-modal strain, and another one for our modal strain. So I'm going to take a tip. Let's see, a good position here. Touch it to a single colony. You don't really need to see anything on the tip. If you see something on the tip, there's a good chance you have too much, and it can inhibit the PCR. So we just take this, put it into the tube, swirl it around, cells come off, put that into waste. Same thing for the non-modal. Touch a colony. So the modal strain has a transposon upstream of the FLHD gene and makes it active. The non-modal does not have a transposon, and we'll be able to tell the difference in the PCR based on the size of the product that we get. All right, so these all go into our cycler, which is already set. Close that. Hit run. Already set to the right volume. Okay, and it will start. And then we'll let that run, and we'll take our samples out when it's done. So I'll give a pause and wait, and we'll come back when that's done. Okay, so the PCR is done. Let's open this up and take our tubes out. And now we're going to add some loading buffer. This just has some glycerol to increase the density and some dyes in it so that we can see where it is. Take that, we can see blue dyes in there. We just mix it up a little bit. The same for the rest. So our samples have run. They have loading buffer in them, and now we're ready to take them and put them into our agarose gel, which we'll talk about in another video.